Okay, and here is our second lecture in the cell, prokaryotes versus eukaryotes. We'll talk about some shared characteristics between the two, and then we'll differentiate them quite a bit, and then we'll go into shapes of bacteria, but also the arrangements. We didn't quite hit on those arrangements. Alright, so as I promised, the shared characteristics of all cells in big old caps is the cell membrane. Okay, that cell membrane is also in all caps because of its importance. Okay, and remember we need to know the chemical structures and functions of these structures too. So remember, the cell membrane is a phospholipid bilayer. Those phospholipids are lipids with the phosphate group, and they are polar hydrophilic on one end and nonpolar hydrophobic on the other end, kind of like this guy. Okay, and then they make a bilayer. Okay, that's the cell membrane in essence, although it is controlled by DNA, so it is more elastic than just a big oil ball. Okay, that oil ball though holds the cytoplasm, so anything in water or just happening in reactions in the cell is in the cytoplasm. It's like a gel-like substance, okay, and it allows kind of like a fluid type matrix, but also like semi-solid for the organelles that we'll talk about. All cells contain DNA. Interestingly, viruses do contain DNA, but some contain RNA. And viruses are not cells, and that is interesting to note. That will be interesting for chapter on viruses. Okay. And then all cells, if they need to do something, they contain ribosomes. And back to viruses, they don't have ribosomes, they're not alive. So all cells that are alive have ribosomes. Those ribosomes make protein. Okay, and that protein is made up of our friend the peptide bond. Okay. And so we both have DNA, both types, prokaryotes and eukaryotes have DNA, so we need to store that DNA. The prokaryotes do not have a nucleus, they lack a nucleus, and are surrounded by a region called a nucleoid. Okay, and that's where their DNA can be found. Eukaryotes instead have a very complex nuclear membrane that we will talk about, and that encloses a nucleus. Okay, and that nucleus is kind of the classic where I put DNA. But it is important to remember that a prokaryote is called a nucleoid, not a nucleus. Okay, there's no membrane-bound area. All right, and then obviously prokaryotes and eukaryotes, as I circled earlier, have a difference in size. You can tell based on their scale what type of cell they would be. Something in the 2 micrometer range is a lot smaller than something in the 40 micrometer range, like we saw. So that size lends them to need to organize all their stuff, all that stuff that they need to function, right? So they are encapsulated in membrane-bound organelles. Okay, the membrane actually folds in on itself, that phospholipid bilayer, and they kind of support these organelles. And organelles just means tiny organs, so cellular organs. In contrast, the prokaryotes have things called inclusions that kind of support where their stuff is to metabolically live and reproduce. Okay, and again, that bigger size lends the eukaryotes to need a cytoskeleton. Okay, that cytoskeleton supports all of our organelles. Okay, and it keeps us fluid and not just kind of sinking with gravity. Okay, so it's an important thing in all eukaryotes, and in fact, an amoeba, the little pseudopods, pseudopoda rather, that come out of an amoeba are actually like disassembling and assembling the cytoskeleton as it goes. It's pretty fun. Prokaryotes are contain a cell membrane, right? All cells contain that cell membrane in big caps, and they contain DNA in a nucleoid region, and they contain ribosomes and a cell wall. Okay, so that cell wall is what separates it from eukaryotes, because not all eukaryotes have a cell wall, but all prokaryotic cells do. Okay, or at least typical ones like archaea might not. Okay, and so some prokaryotic cells also include a flagellum, anapilli, anaphimbriae, and capsules. Okay, and we can kind of go into all of that later. Okay, and again, not all of those are found on each and every prokaryotic cell, just some of them. All right, but what's interesting to note is most of the cells that you find will all share this shape. Okay, it'll either be spherical or coccus, okay, or there'll be a rod shape or bacillus. Okay, so round wouldn't be wrong, but sphere is probably more accurate. Okay, so you can put sphere, you can do rod for bacillus. Okay, but you'll need to be able to say bacillus, and you'll need to be able to say coccus on the lab practical, and you'll need to be able to recognize those throughout the final and this first exam. Vibrio is very important because of Vibrio cholera. Okay, it's got an interesting shape where it's a curved rod. All right, we won't see that in the lab though, but you will need to know that. Okay, and then we have a spirillum and a spirochete just like we did before. What we want to bring up this time is the arrangements that we can have either with bacillus or coccus. Okay, so if you have one bacillus, you have a single rod, but if you have a chain of rods, you are strepto. Okay, so you kind of just have to think of strepto as a chain. Okay, because we see that in just a second with the coccus as well. Okay, and coccus can form a diplococcus, either two of them. Diplo means di, two. Tetrad is tetra, four. And a chain of spheres is strepto also. Okay, so strepto is a chain. Okay, and then staphylococcus is a cluster. Okay, and the cluster will actually look like grapes, especially if it's gram-positive, because they'll look purple spheres and all clustered together. You may have already seen those in lab. All right, so you want to remember streptococcus is a chain of coccus. You want to know streptobacillus is a chain of bacillus, and you want to know that staphylococcus is a cluster of coccus. Okay, and then the diplococcus and the tetrad you'll want to know as well. So take this to memory. You'll see it on the first exam. You'll see it on the final. All right, and then we'll move to osmotic pressure, and I'll see you there.